Hi, hi everyone. I'm delighted to be joined by Professor Mimoun Azzouz, Chair of Translational Neuroscience at the University of Sheffield, founder and director of the Gene Therapy Innovation and Manufacturing Center, and a leading figure in the development of gene therapies for neurological diseases. Over the course of his career, Professor Azzouz has helped advance gene therapy from a promising concept to a clinical reality, particularly in conditions affecting the brain and nervous system. His work spans fundamental neuroscience, translational research, manufacturing innovation, and biotech entrepreneurship. In recognition of his outstanding contributions to the field, he recently received the British Society for Gene and Cell Therapies Outstanding Achievement Award. Professor Azzouz, congratulations on the award, and thank you for joining us today.
Thank you. Thanks, Rebecca. Thanks for inviting me for this interview.
Thank you for making the time for it. And let's start at the beginning. What first drew you into neuroscience and ultimately into gene therapy as a way to treat disease?
Yeah, since May, I was very young. I found that the brain or aspects, it's fascinating to understand, amazing to understand how the brain function and control our body, basically. So I got interested in neuroscience, particularly when I was at school, and then this interest increased when I moved to the university. So I had lectures about the brain, the anatomy, the functions and different neurons. It's amazing to see the various aspects of the brain and the spinal cord. So that's where my interest, I got interested in the brain and the spinal cord and neuroscience overall. And then I started having interest in studying the diseases affecting the brain and spinal cord, and it started with Parkinson's disease, and then ALS, motor neuron during my master and PhD. One thing that really got me interested in gene therapy was originally to see many, in particular young children affected with genetic inherited disorders, and where we know the gene that affects causes the disease or inherited in general. So that's the reason I started reading about gene therapy and looking for career opportunities in gene therapy. That's why I started, working in this field in 1997 in Switzerland.
That's very interesting. And actually, the next question relates a bit to the previous one. I was wondering if you can share an example. What was the state of gene therapy when you first entered the field? So you just mentioned this was in 1997. And what have been the biggest scientific or technical shifts you've witnessed in gene and cell therapy over the course of your career?
Yeah, so when I started gene therapy in Switzerland, in Lausanne, one thing that came up at that time was lentiviral vector. So it was quite a hot area at that time, generating vectors, in particular HIV-based lentiviral vectors. So I started working on that. But obviously, lenti is great for direct delivery, restricted area for gene therapy for restricted area that is affected in a condition. We knew that at that time. It's not spreading if you deliver in the blood or in the CSF for CNS applications. So I started working on AAV at that time, but it was AAV2. So it was really restricted what you can do at that time with these vector systems back in 1997. Another limitation that we faced at the time, it was the first time I started working on spinal muscular atrophy, SMA. And at that time, we knew the gene that was discovered, I think, 2-3 years before I started my PhD, a postdoc working on SMA. But in terms of model systems, in particular, iPSC-derived motor neurons or mouse model at that time it didn't exist. So we had to basically, try to develop therapy with what we had at that time. So that's the key limitation of that time. What changed since then over my career was mainly a wealth of vector systems emerging over the years, starting with improvement in lentiviral vectors. And I was involved in that in Oxford, for example, rabies pseudotyping lentiviral vector, which was very promising. And then various serotypes of AAV that emerged over the years. That made a huge difference in the field of gene therapy. And the stories around vector system based on AAV that made it to the clinic with promising results and then to the market after that. So that's basically the key evolution or progress, breakthroughs in the gene therapy over the years.
Yeah, that's very interesting because we still see that ongoing evolution, particularly with AAVs, where quite often we see these new capsids and chimeric capsids, and still ongoing.
Yeah, still ongoing. Yeah.
Okay, the next question is, can you share an example of a moment in your research where something unexpected, either a setback or a breakthrough, changed the direction of your thinking?
Yeah, I mean, it's a breakthrough, but at the same time, a huge disappointment. An example of gene therapy based on AAV9 for spinal muscular atrophy, semen replacement. So we were really pleased by the breakthrough when we generated proof of concept for gene replacement for SMN replacement for spinal muscular atrophy. That was achieved in 2010. And since then, after that, we were really pleased to secure the funding to take this approach to the clinic, which we started working on. I remember that was an MRC award to help us manufacture and progress it to clinical trial approval at the time. The problem we faced was, and that was a huge disappointment, is that in terms of manufacturing capacity in the UK was not, we had no possibility to manufacture tox material and clinical trial, clinical vector to enter phase 1/2 clinical trial in SMA patients. We spent, nearly 2 years queuing for slots, in the UK, no capacity, so we had to go to the US. But we faced many, many challenges at that time, trying to secure a slot, and for some reason it didn't work because we started working with a few sites, 2 sites there, but in the end we couldn't work with them because the sites were either bought by a pharma company or they had their own priorities. So that was a disappointment because we couldn't progress that promising therapy to the clinic. And you know the story in terms of basically in the US, the same approach was progress to the clinic and to commercialization. So in terms of disappointment for the entire, for us in Sheffield, for the UK, it's a missed opportunity basically.
Yeah, I remember hearing the story in one of your talks recently, and I do have another question coming up later relating to manufacturing. But before we move on to that, the field is advancing rapidly with technologies such as new AAV vectors, as we just mentioned, novel genome editing strategies, and new delivery systems. Where do you think we are currently underestimating what is possible in gene therapies?
I think, some aspects I remember some colleagues saying that, oh, gene editing, for example, is too far away from clinical applications. There are many safety consideration before we apply this technology in human. But in fact, it went really, really fast. And a good example of that is the approval of the CRISPR gene editing approach for sickle cell disease and beta thalassemia. So this is a good example of the fast-moving field of which is gene therapy and including gene editing. So probably we underestimate that we can go faster in this area, but that's a very good example of how great the potential of gene therapy, gene editing, and we can move really, really fast with this technology.
Yeah, I absolutely agree. I did my PhD a few years ago on gene editing, and even a few years ago, it was still a question you will hear often, like, but is this really translational? And now here we are.
Yeah.
On that topic, what emerging technologies or approaches in gene and cell therapy are you most excited about right now? Is there a particular emerging technology that you've heard of recently or are particularly excited about?
Yeah, I mean, there are many exciting, it's not only disappointing news about gene therapy, but there are also some exciting news coming up. In particular, you did mention new capsids emerging. In particular, I'm excited by the new tools that are used to benefit gene therapy technology. We are talking about tools like AI, for example, using AI to engineer for capsid engineering. It's a good example. We can see now some of promising capsids emerging, and hopefully with improved safety profile compared to AAV9, for example. But the field is moving really fast on that front, and these tools are helping not only capsid engineering improved in terms of efficacy and safety. It will help also dosing in patients if we have more efficient capsids. These tools like AI are helping also other aspects like process development, manufacturing, also prediction of safety and efficacy of gene therapy treatment in patients. That's very important, and it's a good example to mention here that basically the regulators are thinking how to apply these technologies like AI in So for the safety, improving the safety, prediction of risks, etc. And yesterday or the day before, and on the 9th June, actually, MHRA announced these new initiatives around using AI in predicting risks and improving safety, which is very, very good. So it's exciting to see the applications of these new tools in gene therapy, and I'm confident that the field will re-emerge from some of the setbacks that we witnessed in the recent years.
Yeah, that's very promising to hear. I think it's quite impressive how quickly our field has adopted AI and integrated it into the science we do.
Absolutely, yeah.
Okay, I'm gonna change the topic a little bit. Translational research is ultimately about the patients. I was wondering if there's a patient story or interaction that has particularly stayed with you during your career, and how important would you consider is patient engagement in shaping research priorities?
Yeah, I think in terms of patients, which is, it's patients are at the heart of what we are doing in gene therapy with our translational programs. Two examples of that really stayed in my memory, and interacting with some parents, in particular mothers of babies affected with SMA, and it's devastating to have a baby and with affected with spinal muscular atrophy, and to see some of the parents, and in particular one mother, just describing the impact, how impactful the treatment that her baby received with Zolgensma, for example, SMN replacement. That was, something that make scientists like myself proud that we try to help patients like babies affected with genetic disorders. Of course, there are many examples as well, when you have parents approaching us, and I'm always pleased to see therapies, discoveries that we make in the labs that progress to clinical trials. And the example of gene therapy approach for spastic paraplegia type 47 is also another example, because at the start of all the efforts were parents that approached us to develop this therapy, and parents of children affected with SPG 47 based in Boston that want to work with us and support all the efforts we did to have an impactful therapy on patients.
Yeah, I think definitely the work that public and patient groups do in our field is pretty impressive. All the effort gathering funding, they have definitely been a fundamental part in moving the field forward, I would say.
That's right, yeah.
Okay, moving on a bit to your research. So a major part of your work focuses on ALS and motor neuron disease. Where do you think we are today in terms of translating gene therapy into meaningful clinical impact for these conditions?
Yeah, I started working on ALS, as I said, motor neuron disease in general, both SMA and ALS back in 1997. Gene therapy, I started working on ALS in 1994. So ALS is basically one of the worst conditions affecting the CNS, probably the worst, and it's a devastating condition. And it's very, very hard to treat as well, because it's fast. After diagnosis, patients, they progress really fast. So our efforts made some interesting discoveries, and that lead to, for example, the development of concepts to target genes that affect some genetic cases of ALS, like SOD1, for example, affecting familial cases of ALS. And the silencing of SOD1, we developed this concept back in 2004, and then progressed with that later on with first with Lenti and AAV. And we are really pleased to see that the same concept is applied using not viral, but using ASOs for oligo ASOs to silence SOD1. And this led to the approval of the first genetic therapy for ALS, which is tofersen. This is great. It demonstrated with some I would say the first remarkable impact on patients with SOD1 mutations. So this is the first example of how impactful these therapies could be when you target the causative factor of the disease, i.e. the mutated gene. And I can see more progress emerging. I think with more genes discovered affecting ALS patients, there will be more genetic applications, gene therapy applications in these patients, both, for example, C9orf related ALS, for example. I am aware some exciting developments ongoing, both using viral and non-viral therapies are coming up.
Thank you. I'm gonna go back a bit to the manufacturing topic. So you founded the Gene Therapy Innovation and Manufacturing Center. What gap was it designed to fill, and how is it changing the way gene therapies are developed in the UK?
Yeah, so I did mention the disappointing outcome with SMN replacement therapy, and that was the driver behind all the efforts that we put in establishing a gene therapy innovation and manufacturing center. Fortunately, MRC and LifeArc, got involved, and they established this call to establish the innovation hubs for gene therapies. One of them was established in Sheffield, the GTIMC, Gene Therapy Innovation and Manufacturing Center. And this hub was established to address mainly two or three shortages. The first one is, as I mentioned, the lack of the low capacity in the UK to manufacture clinical grade vectors, mainly for academic groups. Because fantastic science is happening in the UK, fantastic discoveries, and what we were missing is this capacity to manufacture these vectors to treat patients in phase 1-2 trial. The second shortage was about the skills and training. So small, as you know, in process development and manufacturing in the UK, we had a small pool of talent and skilled individuals in this area. And for academic institutions, sites for manufacturing, to compete with biotech or pharma on this small pool of talent, it was quite challenging. So the hub was established with this idea to have a pillar for training and skills to upskill the next generation of talent. And also a third pillar was around basically translational support. So providing support needed to take discoveries from early stage all the way to clinical trial. And the hub started doing that, so providing support for academic groups, in particular academic groups with less experience in gene therapy field. And GTMC provides support even attending MHRA scientific advice meetings with academic groups to help and support progressing their therapies all the way to clinical applications.
Thank you. Yeah, that has definitely changed the playing field in the UK, I would say. And on that note of the translational pathway, you've co-founded companies including Blackfin Bio and Crucible Therapeutics. What has been your biggest lesson from taking academic discoveries into biotech and the industry space?
The biggest lesson, yeah, I would say there are 2 or 3 learning experience from that. The first one is obviously you need to realize that once the discovery moves out from the university to a startup company, you need to start thinking differently in terms of the way the program is run. And the second aspect is it's quite hard to, in the UK landscape at the moment, and in particular 2025, the investment in startup company was a huge challenge. So you need to, the learning lesson from that is that obviously you spend a lot of time presenting to investors, talking to investors, and you need to move a little bit away from your hat of wearing the hat of academic investigator to more entrepreneur, and start talking to investors about the potential of your company, the potential of your assets, lead asset and other assets in the pipeline of the company. So, but it's exciting experience. It's it's very important experience and path to go through, because what I realize is you make exciting discoveries in the lab, but if there is no clear path to translate them to the clinic and later on to the commercialization, it's really a challenge. So you need to have that path all the way to getting investment and taking the therapy all the way to phase 1 trial, but also beyond pivotal trials and launch into the market.
Yeah, I can imagine it's almost you have to change your mentality a bit when you're about to pitch to investors, and perhaps change what you would focus on as a scientist when trying to get investment for this.
Absolutely.
Thank you. On that topic, what advice would you give to researchers considering commercializing their work?
Yeah, I think there are many opportunities now for young investigators, even at postdoc level and early career scientists, fellows, to be engaged with the what we call the commercialization journey. So, for example, at the University of Sheffield, this opportunity exists. Postdoc, including in my team and fellows, they can engage with this commercialization journey. It's like a training that they receive. So they work on a project, they can take it all the way through the journey, through different stages, with the support from the commercialization team. And that's a great opportunity for them, because they will think about what it is needed to take their research to startup stage or commercialization or licensing out, etc. And that is the advice I give to more ECRs to be involved in this type of activities, and it would be very beneficial for them, giving them the opportunity to even think about IP and patents in advance, and think about also what are the gaps and the gaps to fill before they bring their program or projects ready for commercialization.
Yeah, it's
And
very
the other, yeah, and the other advice, sorry, other advice is basically, obviously, this journey of translational pathway is full of challenges. Absolutely full of challenges. You sort out a challenge, another one emerge at different levels. When you talk about tox studies, manufacturing, thinking about regulatory pathways, regulatory advice, making sure that you align everything to the clinical trial, which is later stage. But also all the challenges, even if you get to the stage where you have approval from regulators to enter clinical trial, there's still a lot of hurdles that you need to go through. But the advice is that you need to seek, you have the right team around you, but also persist, keep going, because by doing that, you will get to achieve your goals and objectives.
Yeah, that's very good to hear. I think that's great advice, and thanks to experienced people like yourself that say these things. It's becoming a bit more common for ECRs to try to engage with their TROs at the universities and start thinking about all these aspects that you just mentioned pretty early on.
Yeah. No, that's right. But that is also going, getting around and addressing some of the errors and mistakes around, because let's say, as academic, we think about submitting abstracts for conferences and publishing papers as soon as possible, but one thing that if you are involved in this commercialization journey, you learn about the importance of making sure you check first that there is nothing you disclose in the abstract or the poster that will impact potential IP filing, for example, and getting yourself in a difficult position with prior art, etc. So, and that's very important.
Absolutely. I was wondering how important are organizations like the British Society for Gene and Cell Therapy in shaping collaboration and accelerating progress in this field, in your opinion? And how can these scientific societies best support the next generations?
Well, these societies like British Society of Gene and Cell Therapy, and this is the same for the European one, the American one, are extremely important for in the field. Important because it's given these societies are now operating widely in terms of not only, for example, organizing conferences, scientific conferences, but also involved in wider, activities, thinking about ECRs, for example, and promoting them and organizing events with them, giving the opportunity for ECRs also to be involved in the society for cell and gene therapy. I think so. So the role of such societies are critical to drive the field forward, and in terms of dissemination, in terms of rising awareness, and also, you know, many activities and tasks that drive the field forward.
Yeah, we are definitely making an effort to support our field, and on that note, I'm gonna make a little advertisement. But we actually, the society, the BSGCT, recently launched a grant for postdocs for ECR. It's a small grant, but it's to generate proof of concept data, which is quite hard to fund at the moment, I would say.
Well, I mean, I think about it even beyond that. So even the non-successful applicants, basically, both successful and non-successful. So what it does is that it's giving them the opportunity to think about writing a grant. So even if it's a 5,000 grant, so it's a learning experience for them to sit down and try to complete the form, think how to structure their grant application. And if they are successful, it's great, because that is going into their CV and it's demonstrating that they take the initiative, they apply to secure income for their research projects. So it is a very, very good, very good initiative from BSGT, I would say.
Thank you. And now we're coming up to our final question. If you look 10 to 15 years ahead, what would you hope gene therapy for neurological diseases will have achieved?
So in the next 10, 15 years.
Yeah.
So what I hope will happen, and I'm confident, I am optimistic. What will happen in the next 10, 15 years is, now with the emerging technologies, both viral and non-viral, I mean, RNA, for example, therapies are emerging quite fast as well. What I'm hoping is that the time from discovery to enter clinical trial will be shortened significantly. So I expect, that is my expectation, is that these therapies will progress faster, and there will be more and more efficient platforms to manufacture and these therapies, and to bring the costs down for some of these expensive therapies. And that's what will happen with all the efforts that are ongoing now to do that, to use tools to improve both discovery, but also manufacturing, clinical design. I expect that with the engagement and alignment with regulators, it's achievable. It's realistic to think in 10, 15 years that these therapies will first progress fast to trials and market faster, and also these therapies will be more efficient, and third, more safe for patients. So that's my expectation and prediction for the next 10, 15 years.
That sounds great. I'm glad it was an optimistic scenario. I love to hear this.
That's based on how fast the field is moving, and but also the full engagement from regulators to make these therapies. A good example is MHRA recently announced some exciting initiatives like rare therapies, and but also the use of AI, etc. So I think the prediction, and there is huge optimism around that.
Thank you so much. And that was my last question. So Professor Azzouz, thank you for sharing your insights and experiences with us. And congratulations once again on receiving the BSGCT Outstanding Achievement Award. Thank you for your time, and thank you everyone for watching.